Your exams don't do flap, neither do we. Straight talk without the sweet talk. All you have to do is listen, read, test, and destroy. Let's start this lecture now.
Now let's talk about microbiology special cultures requirements. When approaching special culture media, the key is to first separate what staining is from what culture media do, because exam writers deliberately test confusion between the two. Stains, gram stain, acid-fast stain, silver stain, etc., are microscopy tools. They tell you about cell wall structure or morphology, but they do not grow organisms. Culture media, on the other hand, are growth environments that supply nutrients or inhibitors to selectively allow certain organisms to survive while others are suppressed. A common tested setup is a patient with classic clinical features where the stem tells you the organism doesn't grow on routine blood agar or needs special factors, pushing you toward a specific medium rather than a stain. Keeping this straight avoids the classic trap of choosing an acid-fast stain when the question is actually asking which medium allows isolation. Haemophilus influenza is the prototype organism for understanding nutritional requirements. It does not grow on standard blood agar because intact red blood cells sequester the necessary growth factors. Instead, it grows on chocolate agar, where RBCs are lysed, releasing factor V, NAD, and factor X, Haemophilus. A classic tested concept is that satellitism occurs when H. influenza grows near Staphylococcus aureus on blood agar because staph releases NAD. This reinforces that chocolate agar is about nutrient availability, not selectivity. Importantly, this is unrelated to staining. H. influenza is gram-negative Coccobacillus on gram stain, but gram stain alone won't help you isolate it. Neisseria gonorrhoeae and Neisseria meningitidis highlight selective media, not enriched media. These organisms are fastidious and easily overgrown by normal flora, so they require Thayer-Martin agar, which contains antibiotics that kill competitors while sparing Neisseria. Vancomycin suppresses gram-positive organisms, colistin and trimethoprim suppress most gram-negative organisms except Neisseria, and nystatin suppresses fungi. A high-yield tested angle is a patient with urethral discharge or meningitis whose specimen is contaminated with normal flora. Growth only occurs on Thayer-Martin, not chocolate alone. This reinforces that Thayer-Martin is selective, whereas chocolate agar is enriched. Gram stain may show kidney-shaped diplococci, but isolation depends on the medium. Bordetella pertussis requires a completely different approach because it is extremely fastidious and sensitive to toxic fatty acids. It grows on Bordet-Gengou agar or Regan-Lowe medium. Bordet-Gengou contains potato extract, while Regan-Lowe contains charcoal, blood, and antibiotics, where charcoal neutralizes toxic substances. A classic teaching point tested indirectly is that the organism produces toxins causing lymphocytosis, but diagnosis early in disease relies on proper culturing. Again, no stain will fix the problem. Failure to grow is due to lack of appropriate media. Corynebacterium diphtheriae uses Tellurite agar or Löffler medium, which enhance identification and toxin production. Tellurite agar causes characteristic colony appearance due to tellurite reduction. The exam trick here is separating diagnosis of toxin-mediated disease from isolation of the organism. The gray pseudomembrane and toxin effects are clinical clues, but culturing requires these specific media. Gram stain alone only shows club-shaped gram-positive rods. Mycobacterium tuberculosis is frequently tested as a slow grower, which immediately distinguishes it from organisms diagnosable by routine culture. It requires Löwenstein-Jensen or Middlebrook medium, and growth may take weeks. Acid-fast staining identifies mycolic acids, but culture is required for confirmation and drug susceptibility. A common test angle contrasts acid-fast stain positivity with slow culture growth, reinforcing that stain and culture answer different diagnostic questions. Mycoplasma pneumoniae demonstrates the extreme end of fastidiousness. Because it lacks a cell wall, it cannot be gram-stained meaningfully and requires Eaton agar, which supplies cholesterol for membrane synthesis. This is often tested by elimination. No growth on blood agar, no gram stain, atypical pneumonia, pointing to a cholesterol-requiring medium. For enteric organisms, McConkey agar is both selective and differential. It inhibits gram-positive organisms and differentiates lactose fermenters, which produce acid and turn colonies pink. This is frequently paired with eosin and methylene blue, EMB, agar, where E. coli produces a green metallic sheen due to strong lactose fermentation. A classic exam distinction is recognizing that both media assess lactose fermentation, but EMB has a visually striking sheen specific to E. coli, while McConkey broadly categorizes lactose fermenters versus non-fermenters. Brucella, Francisella, Legionella, and Pasteurella are grouped together because they require buffered charcoal yeast extract agar supplemented with cysteine and iron. Charcoal neutralizes toxic metabolites, while cysteine and iron support growth. A frequent tested clue is severe pneumonia with hyponatremia or exposure history where routine cultures are negative, pointing toward a charcoal-based medium rather than staining differences. Finally, fungi grow on sabouraud agar, which has an acidic pH that inhibits bacterial growth. This is often contrasted with fungal stains, like silver stains, emphasizing again that stains help visualize organisms, but sabouraud agar is for growth, not identification under microscopy. In summary, the unifying testable framework is, stains visualize structure, culture media enable growth, enriched media supply nutrients, selective media suppress competitors, and differential media distinguish metabolic traits. When a stem emphasizes failure to grow on routine media, think culture requirements. When it emphasizes appearance under the microscope, think staining. Keeping these domains separate and recognizing which organisms demand special handling is the core concept repeatedly assessed. Just to recall now, keeping staining, special stains, and special culture media organized mentally, it helps to lock in their purpose and sequence in diagnosis. Staining is a microscopic visualization tool. It does not grow organisms and cannot rescue organisms that fail to survive outside the host. The Gram stain rapidly categorizes bacteria based on cell wall structure, separating organisms with thick peptidoglycan from those with thin walls and an outer membrane. And it also gives morphology, coxae versus rods, chains versus clusters, which narrows the differential immediately. When organisms do not stain well or have unique cell wall components, special stains are used. Acid-fast staining detects mycolic acid-rich organisms that resist decolorization. Silver stains highlight thin or poorly staining organisms. GIMSA helps visualize intracellular organisms, and India ink highlights capsules by negative staining. These stains answer the question, what does the organism look like and what is its wall made of? But they never answer, can I grow it? Special culture media answer a completely different question. What does this organism need to survive and multiply outside the body? Some organisms require enriched media because routine agar lacks critical nutrients, while others require selective media because they are easily overgrown by normal flora. Chocolate agar supplies released intracellular growth factors. Selective media like Thayer-Martin suppress competing bacteria and fungi. Charcoal-based media neutralize toxic metabolites. Cholesterol-containing media support organisms without cell walls. Acidic media favor fungal growth, and slow-growing organisms require specialized media and extended incubation. A commonly tested diagnostic pattern is that stains may suggest an organism's identity, but failure to grow on routine culture forces you to think of special culture requirements, not a different stain. The organizing rule is simple but repeatedly tested. Stains classify and visualize, special stains compensate for unusual structures, and special culture media provide the environment needed for growth. Mixing these roles is where mistakes are intentionally targeted.